Okay, I promise not to explode in the screen again. The other neurotransmitter that I'd like to discuss actually is one called serotonin, which is another one that most people have actually heard of before or are familiar with. Serotonin is another monoamine neurotransmitter because it, like dopamine and norepinephrine, are manufactured from an amino acid, but in the case of norepinephrine and dopamine, they're manufactured from the same amino acid, right? Tyrosine. Well, serotonin is manufactured from a different amino acid called tryptophan. And while dopamine and norepinephrine share that catechol ring and are catecholamines, dopamine, norepinephrine, and serotonin are not. They don't share that feature. Serotonin is not a catecholamine; it's just a monoamine. Now, serotonin is very important for psychological health and for mood states. Serotonin actually has a great many potential postsynaptic receptor targets. So I should mention that, for example, acetylcholine had the nicotinic, the ionotropic postsynaptic receptors. There are also metabotropic receptors for acetylcholine. They're called muscarinic receptors. And then for dopamine and norepinephrine, there are significant postsynaptic metabotropic receptors. There are about 5, distinct kinds for dopamine, for example. For serotonin, we have identified 15 distinct, potential receptor targets postsynaptically for serotonin. 14 of those are actually metabotropic, linked to different G proteins and different intracellular, biochemical cascades and influences over gene expression and protein synthesis, and one ionotropic one. So there's a huge diversity of potential, responses to the release of serotonin. And serotonin, appears to be very important for sort of psychological health and, again, mood states. Drugs that are used to treat, for example, depression typically target the serotonin system. They tend to, a very popular class of drugs, obviously, are what are called selective, right, serotonin reuptake inhibitors. Now, when I talked earlier about how acetylcholine is released, right, into the cleft, and then it's broken down by a degradative enzyme in the cleft. Well, the monoamines have a very different, sort of process for ending the message, okay? Now, the monoamines are manufactured inside, the presynaptic terminal, the same way that acetylcholine was, right, by synthesizing enzymes, and they are packaged into vesicles. So there are, vesicular transporters that are, sit in the vesicle membrane, right, and pull in dopamine or norepinephrine or serotonin, whichever, neurotransmitter is going to be released from that presynaptic terminal. And then they are, of course, also released into the cleft upon the arrival of an action potential. But the message for the monoamines is ended in a very different way. It's not broken down by an enzyme in the synaptic cleft. What happens first for these monoamines is there is another transporter. It's a selective transporter. It has a nozzle that's shaped either like dopamine or like norepinephrine or like serotonin, and it sits in the presynaptic terminal membrane. So if you're thinking of our presynaptic terminal with the vesicles, right, and they all release serotonin, let's say, into the cleft, well, right on that vesicle membrane right here, you're going to have proteins embedded in that membrane, and they're going to be transporters. So they're going to literally use ATP to suck the serotonin like right out of the cleft after it's been released. And then once the serotonin, or in the case of a dopamine synapse, once the dopamine, or in the case of a norepinephrine synapse, once the norepinephrine comes back inside that presynaptic terminal, there's an enzyme, another kind of protein, right? It's a degradative enzyme. It doesn't care what, which monoamine it goes after, but it will break down, any of the monoamines that it finds. So it breaks down, norepinephrine or serotonin or dopamine. It's called monoamine oxidase, or MAO, right? MAO is the enzyme found inside the presynaptic terminal that'll grab onto any monoamine and break it down. So in terms of ending the message of a monoamine, dopamine, serotonin, norepinephrine, they're released into the cleft. They are first sucked back in to the presynaptic terminal via a selective transporter, selective for serotonin, selective for dopamine, selective for norepinephrine, and then inside that presynaptic terminal, MAO, monoamine oxidase, the degradative enzyme, grabs them and breaks them down. Well, to get back to the original point, for serotonin is typically, the target of a lot of drugs that are designed to treat, depression. And what they do is like Prozac, for example, is what we call a selective serotonin reuptake inhibitor. Well, the process by which the monoamine, let's say serotonin in this case, is taken back into the presynaptic terminal, pulled back in. It's done through this transporter, right? This particular machine. This particular machine transporter is called a reuptake transporter, and this is proof that scientists are not English majors, because when they say what's happening here is the serotonin is being reuptaken, from the cleft into the presynaptic terminal, and the reuptake transporter is the protein machine that uses ATP to sort of suck it back out and back in, to the presynaptic terminal. Well, Prozac and these other SSRIs selectively block the reuptake transporters for serotonin. Now think about what happens if you block, the mechanism by which you remove serotonin from the cleft. Well, when you get serotonin released, upon the arrival of an action potential, the message doesn't end. It lingers in that synaptic cleft and starts stimulating those, very complex, arrays of, up to 15 distinct, postsynaptic receptors for serotonin, and activating all sorts of G proteins and all sorts of biochemical cascades. So the interesting thing about these drugs like Prozac and SSRIs that actually act to relieve depression, the symptoms of depression, is that they don't work instantaneously. They don't work right away, the first time you take them. They need to, it takes a couple of weeks, actually, typically, for enough change to occur in how your neurons are wired up to actually generate, relief from the symptoms of depression. And this really does suggest that actual physical growth in the structure of these networks is occurring and needs to occur in order to relieve the symptoms of depression.